Welcome to the first lecture in a series of lectures dealing with modifications in basic principles. And really what we're referring to as modifications are changes or alterations in phenotypic outcome different than what we would expect from a classic Mendelian dominant recessive relationship. So first, let's review a few things before we continue on with today's lecture. In the past lectures, we discussed Mendel's work and the hypotheses that he proposed to describe the phenomena that he saw in his research and his analysis. One thing that Mendel proposed was that unit factors occurred in pairs. Now, unit factors or characters are things that Mendel referred to as distinct heritable features. He proposed that these unit factors occurred in pairs, more specifically that genetic characteristics are controlled by unit factors that exist in pairs in individual organisms. He also then went on to propose the idea which is known as the principle of dominance and recessiveness. In this example, what he proposed was that if an individual had two unit factors that were distinct from one another, one of those unit factors was dominant and was expressed or seen in the phenotype of the offspring, and the other one was recessive or basically masked or not seen in the phenotype of the offspring. He also contributed the idea of the law of segregation. What he proposed here was that during gamete formation, the paired unit factors segregate or separate randomly so that each gamete receives one copy or the other. And then lastly, he proposed the law of independent assortment. And this states that during gamete formation, segregating pairs of unit factors assort independently of each other, meaning that one specific unit factor separated distinctly or separately from another unit factor. So there was no bearing of getting one associated with the other. So again, Mendel proposed this concept of a unit factor or a character. And again, he described this as a distinct heritable feature. In our modern day terminology, we can use the term gene to represent what Mendel meant by a unit factor or a character. And a gene in our more modern day terminology is a heritable factor that occupies a specific chromosomal location. In addition to that, Mendel used the term trait. Now, when he was talking about a trait, what he referred to was different variants or variations to a given character. So this table on the right is one that we saw or looked at in the previous lecture, and really what it does is just identify some of the characteristics that Mendel was looking at in his studies. But more importantly, what it helps us do is differentiate the concept of a character or unit factor, or more specifically again with modern day terminology, a gene, from a trait, which in our modern day terminology is referred to as an allele. And an allele is an alternative form of a given gene. So again, referring to the table, what you can see across the top is what we might refer to as the character or the unit factor involved or the gene. We might have a gene for seed shape, or we might have a gene for seed color, or for flower color, or for pod shape, or for pod color, and so on. More specifically, there are alternative forms of that gene or genetic information. So for seed shape, in Mendel's case, there were two options. Seeds could either be round or they could be wrinkled. And this refers to the different alleles that are present. They are slightly different sets of information. They are still genes that code for shape, but there is genetic information that would cause the shape to be round versus genetic information that would cause the gene to be wrinkled. So again, I just want to clarify and make sure that the concept between a gene and an allele is very clear. Now, when we focus on an allele, there's some additional terminology that we can utilize. Again, an allele is described or defined as alternative forms of a gene. You can have something that's referred to as the wild-type allele. This is the allele that occurs most frequently in a population and is oftentimes arbitrarily labeled or tagged as quote-unquote normal. Usually this is dominant, but not necessarily always the case. In addition, you can see the terminology or utilize the terminology mutant allele. A mutant allele is modified in its genetic information and oftentimes refers or results in an altered gene product. We will talk about mutations later on in the semester. Mutations in the genetic sense are not necessarily bad. Oftentimes we have that connotation with the word mutant that it's something bad, but it's not necessarily something bad. In the genetic sense, it really just means an alteration in genetic sequence compared to a reference sequence, such as the wild type. So a mutant would just be a sequence that is different in some way from the wild type. And the last thing I want to touch on, which I discussed a little bit in the previous lecture, is the concept of what is the convention for gene symbols or allelic symbols. What is normally done is to take the first letter of the name of the recessive trait, and for that you would use a lowercase letter to indicate the recessive allele and the same letter in uppercase to indicate the dominant allele. So going back to those examples that we went over and over again with Mendel, specifically the dwarf versus the tall pea plant, in that particular case, if you note, I utilized the letter D. And the reason I used the letter D was D is the first letter from the recessive trait, which was dwarf, and uppercase indicated dominant and lowercase indicated recessive. That's important to understand that that's for classic dominant recessive relationships. What today's lecture is going to jump into is modifications in phenotypic outcome that are different from the classic dominant recessive relationship that we just established in the previous lectures. Now, before we jump into these modification of basic principles, again, modifications in phenotypic outcome from what we would expect, what I want to do is, again, just reiterate that the Mendelian patterns of independent assortment as well as segregation are going to hold true even for these modifications, again, in phenotypic outcome. So the first example is seen on this slide. It's referred to as incomplete dominance. With incomplete dominance, the F1 hybrids, that means the F1 offspring, have an appearance that is in between the phenotypes of two parents involved in the cross. So let's look at the example on the right. What you see in the parental generation is a red snapdragon flower that is true breeding for the red color, and a white snapdragon flower, which is true breeding for the white color. We know from those basic principles that we discussed previously, as far as segregation was concerned, that the only type of genetic information that the red flower could pass on in its gametes is the red allele, and the only genetic information that the white parent could pass on is the white allele. So then if you look at the F1 generation, what you see is that the F1 are all heterozygous, exactly what we would expect from the Mendelian principles we talked about last time. However, here what you're seeing is a phenotype that is different than what you would expect. In a classic dominant recessive relationship, you would expect to see one of the traits as the dominant trait and the other as the recessive being masked. However, in this case, what we're seeing are pink flowers in the heterozygous individuals. This almost looks like blending inheritance, which Mendel's concepts did not support. He proposed the idea of particulate inheritance. So let's take a look and see what's going on here. If you continue on to take individuals from the F1, again, what we would expect due to segregation is that half of the gametes would contain the red allele and half of the gametes would contain the white allele. If you continue on to make a Punnett square and make a prediction about the offspring that we would expect in the F2, you would expect that 1/4 of the offspring would be homozygous red, 2/4 of the offspring heterozygous, and 1/4 of the offspring homozygous white. But again, in this case, with incomplete dominance, what we're seeing is heterozygotes have a phenotype that is somewhere in between either of the true breeding parents. So the key here to understand what is happening in this modification in phenotypic outcome is due to what's happening biochemically in these individuals. It turns out that the white allele is actually a mutation that doesn't allow for any pigment to be produced. So when you have an individual that has 2 copies of the red allele, they have 2 alleles that are instructions to make the red pigment, whereas individuals that are homozygous for the white allele have 2 sets of instructions, 2 alleles, that basically do not allow for pigment to be produced at all because of the mutation. So when you look at the heterozygous individuals have 1 allele that are instructions to make red pigment and 1 allele that are instructions to make no pigment at all. These individuals have half as much red pigment compared to individuals that are homozygous red. Or you can flip that around and say homozygous red individuals have twice as much red pigment than those that are heterozygous, all based on the genetic information that is present. Now, the other thing I want to highlight here is that the convention changes just a little bit when talking about incomplete dominance and letter designations for alleles. Because it's not a true dominant recessive relationship, we don't want to use uppercase and lowercase letters because, again, that does imply classic dominant recessive relationships. So the convention changes a bit to use a uppercase letter designation, and here I've used the letter C for color of the flower, with a superscript R for the red allele or a superscript W for the white allele. So if you look at the genotypes possible, you can have homozygous red, you can have heterozygous, and you can have homozygous white. So this information is actually the correct genetic way to indicate alleles in incomplete dominant situations. Even though the figure on the right has the classic uppercase, lowercase letters, that's incorrect. And so I've modified that just a bit in this slide on the left-hand side. Here is another example. This happens to be an example in humans. In this case, the disorder that we're talking about is something known as hypercholesterolemia. This basically is a situation where there are very high levels, dangerously high levels of cholesterol in the blood of the individual afflicted with this particular disorder. It is due to incomplete dominance. What you can see with the genotypes listed below, as well as the phenotypes, is what's happening in these situations. So if you look at a homozygous individual that is able to produce a particular receptor, those individuals are phenotypically normal. On the far right, you have individuals that are homozygous for the mutation, and in this case, they don't produce the receptor that helps draw in the cholesterol from the blood, and so these individuals have a very severe form of this disorder. In the center, you have heterozygous individuals, and heterozygous individuals have 1 allele that allows for the production of the receptor and 1 allele that does not. So in the figure, you can see that these individuals do have some of the receptors, but the receptors, more specifically, are about half as much of the normal receptors that you would find as far as quantity or concentration is concerned. And these individuals have a mild form of the disease. They do have a receptor that can work, but they don't have enough of the receptor overall. The next example in modification of phenotypic outcome is a situation known as codominance. Codominance is a condition in which the phenotype or the phenotypic effects of a gene's alleles are fully and simultaneously expressed in the heterozygote. So this again is a situation that would arise in heterozygous individuals. Specifically in this case, both alleles are fully expressed and seen in the offspring overall. So the example of this is what's known as the MN blood group. In this particular situation, red blood cells contain a transmembrane glycoprotein that there are two different forms known to exist, the M form and the N form. First, you can see the different allelic designations. Again, I'm using the letter with a superscript to indicate the allele. So the capital L with the superscript M indicates the M allele, and the capital letter L with the superscript N indicates the N allele. In this case, you can look at the genotypes, and the genotypes would be homozygous for M, heterozygous M and N, and homozygous N. In this case, again, it's specific to the heterozygous individuals. In this case, these individuals express both the M form and the N form. It's distinct from the example we just looked at from incomplete dominance, because in this case, both copies, both alleles are fully expressed and seen in the offspring. The next modification I want to talk a little bit about is the concept of multiple alleles. It turns out that everything we've talked about so far has two possible allelic versions. So, for example, we talked about dwarf or tall plants, or yellow or green peas. There were only two alleles possible. And in the last examples, even with those modifications, we talked about two alleles possible. M versus N allele for codominant example. And then we also talked about the red versus the white allele in the flower example for incomplete dominance. But in nature, it turns out that it's not necessarily always such a straightforward situation where there are only two alleles possible. In many cases, for many genes, multiple alleles can or have been determined or detected or identified. So there is a table in your book, and I apologize if it is not table 4.1, but there is a table in your book that demonstrates an extreme case of multiple alleles, and that there have been over 100 alleles identified for a given genetic location in Drosophila. And so you can imagine that with over 100 different alleles, there are lots of different genotypic outcomes and phenotypic outcomes that you would expect depending upon the relationship of the two alleles in an individual in question. So that's a really extreme example. Let's take that down a few notches and look at a little more simple example by simply just adding one more allele. So again, so far we've looked at two alleles. Let's add one more in a 3-allele system. And the classic example of that is the ABO blood group in humans. Now, the ABO blood group in humans is characterized by the presence of a glycoprotein that is on the surface of the red blood cell. This is distinct from the M and N blood group we just talked about. It is the blood grouping that, if you know your blood type, this is where that comes from, is this particular glycoprotein. It is interesting in that there are 3 alleles. There is the A allele, the B allele, and something known as the O allele. Interestingly, this particular example also exhibits codominance and incomplete dominance. So let's go ahead and look at the table to try and show you or depict some of that. So let's first look at genotype. In the first row for genotypes, you have homozygous A. Well, that's no question. Those individuals would have the A form of the protein. on the surface of their red blood cell, and their blood type would be type A. But individuals that are heterozygous AO, and I apologize here too, unfortunately for the blood typing, the conventional nomenclature changes just a little bit. Here they use the lowercase I for this recessive O allele, rather than what I would use would be the capital letter I with a superscript O. But that's the convention for this particular example, unfortunately. So back to heterozygous individuals that have one copy of the A allele and one copy of the O allele. In this case, these individuals have the blood group or blood type A. So both homozygous A and heterozygous AO display the A form of the protein on the surface of the red blood cell. We can then move down to blood type B, look at those genotypes. Homozygous B individuals, again, no surprise, display or express the B form of the protein. And also individuals that are heterozygous BO also display or express the B form of the protein. Individuals in the next row that have the blood type AB are heterozygous individuals, and this is another example of codominance. With this example, both the A form and the B form are expressed. Both types of proteins are found on the surface of these individuals' red blood cells. Lastly, you have at the bottom, with the last row, is individuals that are type O blood. They have the genotype homozygous O, and these individuals don't have this particular protein on the surface of their red blood cell. So in this case, this is another example where the O allele is a mutation that results in this particular protein not being produced at all. So that then explains why individuals that are heterozygous AO or individuals that are heterozygous BO have either the A protein on the surface of their red blood cell or the B protein on the surface of their red blood cell, respectively. This is a case of incomplete dominance in those examples. The next example in modifications of phenotypic outcomes is what's referred to as lethal alleles. And it's exactly what their name implies. These are alleles that in some form or another cause lethality or death of the individual. So in this classic example, what we have is an allele. We have two different alleles. So we've got the capital letter A, which is for coat color in mice, and it's referred to as agouti. And if you look at the bottom left-hand corner, you see what an agouti mouse looks like. They have basically banded or striped hairs, and they have this kind of brownish sort of color associated with them. The second allele for coat color is indicated with the A and a superscript Y. In this case, this results in a yellow mouse, which is on the right-hand bottom corner. And, we might argue that it's not necessarily yellow, but it's definitely distinct in color from the agouti mouse. In this case, this particular allele acts in a way to be dominant for color, but if there are two copies of this allele present, results in lethality. So let's work through the examples. In cross A, if you have two individuals that are homozygous agouti, there's no question here that the offspring produced would be also agouti and all survive in this particular case. In cross B, what you can see is if you have an individual who is heterozygous in genotype, but yellow in phenotype, are crossed with one another, the outcome that you would expect would be 1/4 homozygous agouti, 2/4 heterozygous yellow, and 1/4 homozygous yellow. However, you can see that the numbers have been altered underneath that modified Punnett square, and that's because in the homozygous yellow genotype, that results in death of the individual before they're actually born. So when you have the offspring that are being produced, what you would have to do is sort of recalculate your phenotypic outcome because you only have the survivors that you can assess or analyze. So in that case, we would now cross out the homozygous yellow individual because they've died, and that leaves us with 2/3 of the offspring being heterozygous and yellow, and 1/3 of the offspring being agouti. If you look at cross-C, this is just another way to expand this concept, in that if you take a homozygous agouti individual with a heterozygous yellow individual, the offspring would be 1/2 agouti and 1/2 yellow, and all would survive, because in this case, none of the offspring would receive 2 copies of the recessive allele. So again, for color, yellow is dominant, because in the heterozygous situation, these individual mice are yellow. However, for lethality, it's the homozygous combination of the yellow allele that causes lethality. It turns out that the mutation is actually in a gene for an RNA binding protein that happens to be right next to the gene that is playing a role in coat color. So with coat color, it causes a modification in the pigment that's being produced, and having only 1 copy of the RNA binding protein defective is still okay and allows for mice to survive. However, if the mice have 2 alleles that are yellow, that also means there are 2 alleles for the RNA binding protein are going to cause death of the individual. Now, oftentimes I get asked, can we use some of those principles that we discussed in the previous lecture for making predictions about the offspring when we have these modifications in phenotypic outcome? And the answer to that is yes. As long as you know what the modification to the phenotypic outcome is or results in, you can still make predictions, because those principles of how the alleles are going to separate and segregate from one another still hold true. So let's look at this particular example. In this particular example, we are going to look at classic dominant recessive relationship for albinism, indicated with the letter A. And we're also going to look at the blood type of the individual. So this is a dihybrid cross. In this cross, we have 2 individuals. Both of the individuals are heterozygous for albinism and heterozygous AB for blood type. What you can do is work them individually, again looking at single monohybrid crosses, and make your predictions of the outcome. So for a classic dominant recessive relationship where 2 individuals that are heterozygous are crossed, we would expect for phenotypic outcome 3/4 of the offspring to be normal pigmentation and 1/4 of the offspring to be albino. If you looked at just blood type alone, again, we're crossing 2 individuals that are both heterozygous AB. In this case, this is one of those modified outcomes in phenotype where we know the AB individuals express both A and B forms of the protein and hence have a different phenotype. So what we would expect from that cross alone would be 1/4 of the offspring to have the A blood type, 2/4 of the offspring to be type AB, and 1/4 of the offspring to be type B. Using our forked line method or branch method, we can put that all back together again and make predictions about the offspring in this cross. If we wanted to know what is the probability that an individual offspring would be pigmented and have type A blood, well, that's 3/4 times 1/4, giving us 3/16. But what if we wanted to know what the probability was of the offspring being albino and type B blood? Well, that's 1/4 albino times 1/4 type B, which also gives us 1/16 albino type B. So again, you can still use the forked line or branched method as long as you know the outcome of the modification that you're dealing with. The next modification in phenotypic outcome that I want to discuss is referred to as epistasis. In epistasis, the word itself comes from the Greek root stoppage. This is where you have an interaction between gene products such that one gene product interferes or prevents with the expression of the other. So the key here is that this is a negative type of interaction. One gene product interferes or prevents the expression of the other. So the first example I want to give you is in Drosophila, which is the common fruit fly. And in this case, we're going to talk about two different sets of genes. One gene is specifically involved in producing the color of the eye in Drosophila. And the second gene in question is a gene that controls the development of the eye. For the eye development gene, there is a mutation that is known as Alleles, and in the homozygous recessive genotype, these individuals do not develop eyes. So, in this particular case, this is an example of epistasis. If you have a Drosophila that is homozygous recessive for Alleles, they do not have any eyes. And because they don't have any eyes, it doesn't matter what their genetic makeup or genotype is for eye color because they don't have any eyes. Again, that's a pretty dramatic example. Another example is the one that is seen here on this particular slide. Again, we're talking about two different sets of genes and their products interacting in a way that one will negatively affect the expression of the other. In this example, we're looking at mice, and we're looking at a gene that is involved in the pigmentation of the hair. Here you have two alleles, black and brown, classic dominant recessive relationship where black is dominant to brown. The second gene in this example is one that is responsible for allowing the pigment to actually be deposited in the hair. This too is a classic dominant recessive relationship where the dominant allows for pigment deposition, so the color to actually go into the hair itself, or the recessive, which is the absence, which does not allow for the pigment to go into the hair. Classic dominant recessive relationship, so the only way you will have a colorless mouse, hence a white mouse, or the absence of pigmentation is in the homozygous recessive genotype. So if you look at the Punnett square on the left-hand side, here we have a cross between two mice that are heterozygous for both color and for this pigmentation gene. If you work through the Punnett square, you can see that the offspring have some interesting phenotypes. In situations where the second gene in question is either homozygous dominant for color deposition or heterozygous, then the color of the mouse is truly dependent upon the genotype for hair color. However, in the bottom of that Punnett square, you can see a few mice that end up being white. These mice are white because they are homozygous recessive for that mutation in the pigment deposition gene. In this particular case, it doesn't matter what their genotype is for hair color if they are homozygous recessive for this deposition mutation gene. Hence, the phenotypic outcome is different than what you would expect in a classic dominant recessive relationship. In this case, following those Mendelian patterns we talked about, in phenotype, we would expect the 9:3 to 3:1 ratio. However, that's not what we see in this example.